Hello students, I am teacher Svetlana and in our today's lecture we will learn about the methodology for rDNA technology, applications of biotechnology in the field of healthcare, agriculture and gene therapy, which is covered in the chapter Biotechnology. So let's begin. Students, let us have a look at the steps that are involved in gene cloning, which are as follows. The first step, isolation of the DNA, that is the gene, from the donor organism. The desired gene to be cloned has to be obtained from the source organism, which is called as a donor. Initially, the cells of the donor organism are shredded with the blender and treated with suitable detergent. Genetic material from the donor is removed, isolated and purified by using several techniques. Isolated DNA can be spooled onto a glass rod. Then the isolated purified DNA is cleaved by using restriction enzymes, particularly restriction endonucleases, which is your ER. These enzymes cleave DNA at specific sites called as restriction sites and break the DNA into fragments. There are several types of restriction endonucleases. Cleaved DNA fragment has cohesive or sticky, staggered ends or blunt ends. Have a look at this image here. This is your plasmid vector that is to be used, and then you have this human insulin gene. Now this human insulin gene is obtained from the donor DNA, and restriction enzymes work on the plasmid, and here site is created where this human gene, that is human insulin gene, can be inserted. So here there are sticky ends or either blunt ends that are formed where the desired gene can be inserted. From the cleaved DNA fragments, a fragment containing the desired gene is isolated and selected for cloning. This is now called as the foreign DNA or the passenger DNA. So this green color insulin gene is your foreign DNA or the passenger DNA. A desired gene can also be obtained directly from the genome library or the complementary DNA library. Then the next step is insertion of the desired foreign gene into a cloning vector, that is your vehicle DNA. The foreign DNA or the passenger DNA is now inserted into the cloning vector or the vehicle DNA. This is your foreign DNA and this is your cloning vector. So it has complementary sticky ends that are present. The most commonly used cloning vectors are plasmids of bacteria and the bacteriophage viruses like lambdaphage and M13. The most commonly used plasmid is PBr322. Plasmids are isolated from the vector organisms, that is the bacterium, by using the same restriction endonuclease which is used in the isolation of the desired gene from the donor. Plasmid, that is the vector DNA, is cleaved. So here also same restriction enzyme is used to make a cut into your plasmid DNA. Now by using the enzyme DNA ligase, foreign DNA is inserted, that is it is integrated into the vector DNA. Can you see here? This foreign DNA has been integrated into the vector DNA by the enzyme DNA ligase. The combination of the vector DNA and the foreign DNA is now called as the recombinant DNA or the chimeric DNA, and the technology is referred as the rDNA technology. So here you have created a recombinant plasmid. Then the third step is the transfer of the recombinant DNA into suitable competent host or a cloning organism. Finally, the recombinant DNA is now introduced, that is transferred for expression into a competent host cell of the suitable cloning organism, usually is a bacterium. Host cell takes up the naked rDNA by the process of transformation and incorporates it into its own chromosomal DNA, which finally expresses the trait controlled by the passenger DNA. The transfer of rDNA into a bacterial cell is assisted by a divalent calcium. Here, have a look at this image here. This is your recombinant DNA that you have. This recombinant DNA is introduced into the bacterium by a process called as a transformation. This is a competent host cell which allows the entry of your recombinant DNA within it, and it gets integrated with the chromosomes and can be then used for the expression of the passenger or the donor gene. The cloning organisms used in plant biotechnology are E. coli and Agrobacterium tumefaciens. The host, that is a competent cell which has taken up the rDNA, is now called as a transformed cell. So this here is a transformed cell. Foreign DNA can also be transferred directly into the naked cells or the protoplasm of the competent host cell without using vector. This is done by using techniques like electroporations, microinjections, lipoinfections, shotguns, ultrasonifications, biolistic methods, etc. But in plant biotechnology, the transformation is through a TI plasmid of Agrobacterium tumefaciens. So you can see here this donor gene can then be, with the process of transcription and translation, the required protein can be produced. Students, the fourth step is selection of the transformed host cell. The transformation process generates a mixed population of transformed, that is the recombinant, and non-transformed, that is non-recombinant host cells. For isolations of the recombinant cells from the non-recombinant cells, marker genes of the plasmid vectors are employed. For example, PBR322 plasmid vector consists different marker genes. Example, ampicillin resistant gene and tetracycline resistant genes. When PST1 restriction endonuclease is used, it knocks out the ampicillin resistant gene from the plasmid so that the recombinant cell becomes sensitive to ampicillin. So here you can see you have the recombinant plasmid, then the transformed recombinant plasmid is present in the E. coli. Then here you have the plate cells on the agarose gel containing ampicillin bacteria. So expand the positive clones to exponentially replicate copies of the desirable gene. So here the selection of the recombinant bacteria is done, and then they are allowed to multiply so that they can produce multiple copies of the desirable gene. The fifth step is multiplication of the transformed host cell. Once transformed, host cells are separated by the screening process. In this step, the transformed host cells are introduced into fresh culture media. At this stage, the host cell divides and redivides along with the replication of the recombinant DNA carried by them. So this step is happening here, where you are allow the cells to multiply. So when the cell is multiplying, it is also multiplying the recombinant DNA along with it. And then the final step is the expression of the gene to obtain the desirable product. The next step involves the productions of desired products like the alcohol, enzymes, antibiotics, etc. Finally, the desired product is separated and purified through downstream processing using suitable bioreactors. So the end step is the protein harvesting. The required or the desirable products are produced and they are just separated by different purification techniques. Now students, let us have a look at the application of biotechnology. Biotechnology is an umbrella term covering a broad spectrum of scientific applications used in many sectors such as health and agriculture, industry, environment, and genomics. Here is a schematic representation of the different fields where there is application of biotechnology. The first application is healthcare biotechnology. It refers to a medical or diagnostic product or a vaccine. This technology has a tremendous impact on meeting the needs of patients. Biotechnology offers patients a variety of new solutions such as unique, targeted and personalized therapeutics and diagnostic solutions for organ transplant, stem cell technology, genetic counseling, forensic medicine, gene probes, genetic fingerprinting and karyotyping are the outcomes of biotechnology. The first immediate application is the human insulin. Insulin is a peptide hormone produced by the beta cells of the islets of Langerhans of the pancreas. It was discovered by Sir Edward Sharpie Scaffer in 1916 while studying islets of Langerhans. Insulin is essential for the control of blood sugar levels. Chemically synthesized DNA sequence of insulin for two chains A and B are separately inserted into two PBR322 plasmid vectors. Insulin production by recombinant DNA technology is designed by Gilbert and Vilokormov in 1978. The genes are inserted by the side of beta-galactosidase gene of the plasmid. The recombinant plasmids were then separately transformed in E. coli host. The host produces penicillinase or plus pre-proinsulin. Insulin is later separated by trypsin treatment. This is the human pancreatic cell, or this is the DNA, and here you have the human insulin producing gene, and on the other hand you have the bacterium which is having the plasmid DNA. This plasmid DNA is removed out, and with the process of recombination you have a recombinant DNA that is formed which has the human insulin producing gene. Then it is further taken and this plasmid is allowed to enter into the bacterium. So we have the recombinant bacterium which is allowed to multiply and produce the human insulin in the fermentation tanks. So once the insulin is produced, it is extracted and purified, and after the purification process the human insulin is ready for human use. Next, in healthcare, we have vaccine production. A vaccine is a biological preparation that provides active acquired immunity against a certain disease. Usually, a vaccine consists of biological agents that represents the disease-causing microorganisms. It is often made from a weakened or killed form of the microorganism, its toxin, or one of its surface protein agents. Now, biotechnology has made advancements in vaccines or vaccinations by making recombinant vaccines that have the potential to eradicate non-communicable diseases like cancer. Naked DNA vaccines, viral vector vaccines, and plant-derived vaccines are found to be more effective against a number of bacterial and viral disorders. We also have oral vaccines, which is a novel approach. The latest hotspot in the field of vaccine research is the development of vaccines which can be taken orally. Immunogenic proteins of certain pathogens is found to be active when administered orally. We also have edible vaccines which are being used. Now, the advantage of these vaccines is there is a comfort of administration, low cost, and ease of storage. This is how this flowchart shows you how the vaccines is produced. You can pause the video for a while and have a look at it. And these are the different oral vaccines. Then the application of biotechnology in agriculture. It involves scientific techniques such as genetically modified organisms, Bt cotton, pest-resistant plant. So it helps in modifying plants, animals, and microorganisms and improve their agricultural productivity. Tissue culture is used in micropropagation, that is large-scale propagation of plants in very short durations. Tissue culture technique is also the best method for storing germplasm and maintaining specific genetic types. This shows the application of biotechnology in agriculture. Then we have gene therapy. A gene is a stretch of DNA required to make a functional product such as part or all of a protein. During gene therapy, the DNA that codes for specific genes is delivered to individual cells into the body. Gene therapy is the treatment of disease by replacing, altering, or supplementing a gene that is absent or abnormal, and those absent or abnormalities which are responsible for the disease. Here is a list of ways in which gene therapy is being used, but the basic principle of the therapy lies: you take the naked DNA, that is the viral vector, the growth factor, which is then introduced into the target cell, and the therapeutic proteins are produced. We have two forms of gene therapy: germline gene therapy, where the healthy gene is introduced into the germ cell like the sperms, egg, or the early embryos, and somatic cell therapy, where the gene is introduced into the somatic cells. Here it allows the transmission of the modified genetic information to the next generation, and here only the affected person is treated and the modified chromosome cannot be passed to the future generation. Thank you, students, for participating in the lecture.